three main questions to really understand proteins and to focus your revision on these three questions even after we're done with this video. Okay? Question 1, how do we describe the structure of proteins? Question 2, what are the roles of proteins? What jobs do they perform in an organism? And 3, how can we investigate proteins practically? Okay? Proteins have, you know it, 4 main levels of structure. Okay? Essentially, that's the primary structure, the secondary structure, the tertiary structure, and the quaternary structure. Okay, they don't always have that, but it is an important part of many proteins. The primary structure is essentially the sequence of amino acids. What are proteins made of? They are polymers, and the monomer of a protein is the amino acid. Okay, so think to yourself how that parallels with the other molecules. Okay, for proteins which are polymers, the monomer, the repeating unit, is an amino acid, but it's not the same amino acid over and over again. Okay, there are 20 different types of amino acids. What's the basic structure of an amino acid? Well, we have the central carbon, we have a carboxylic acid group on one side, we have an amino group on the other side, or an amine group, we have a hydrogen down below, and we have a variable group which can be 20 different things to form the 20 building blocks of proteins, the 20 amino acids. Okay, so this R group can be variable, and there are 20 different varieties. Okay, now what happens to form the polymer? So we got these amino acids, that's great. How does the polymer form? It forms from condensation reactions. So what happens is that the two amino acids so arranged, what will happen is this oxygen and these two hydrogens combine to form the water. It's a repeating story, okay? And as a result of the loss of those atoms, the carbon, instead of unable now to form a bond to that oxygen, it forms a bond to that nitrogen from the adjacent amino acid, forming the peptide bond. Okay, so we get the water formation, H2O, and we get the carbon bonded covalently to that nitrogen, forming a peptide bond. Via condensation reaction, dipeptide formed, water also formed, and the same thing can happen at the next amino acid, and so on until we get a chain of amino acids. This chain of amino acids, so let's just note down the key information here. The condensation results in the formation of peptide bonds. And you can form a dipeptide and a tripeptide and a tetrapeptide and so on until you form a polypeptide, a chain of many amino acids joined to each other by peptide bonds. Okay? However, there are 20 different types of amino acid, okay? And it's the sequence of these 20 amino acids in a polypeptide that makes up the first level of a protein's identity and shape and function, okay? So, the sequence of amino acids, it is the sequence of the 20 amino acids, it's the sequence of amino acids in a polypeptide that is its primary structure. And every different type of protein will have a unique primary structure. Okay? And because of that, the further levels of the protein structure then similarly are unique as well. Secondary structure, so we've already got now our beads on a string. The secondary structure is simply the hydrogen, the folding of the chain by hydrogen bonding. Okay? So what then happens, because the peptide group, in the formation of a peptide group, we have partial positive and partial negative bits. Okay? What do I mean by that? So this is what peptide bond looks like. Okay? This is the thing that connects every amino acid. Now, that oxygen is partially negative, and that hydrogen is partially positive. And because of that, and this nitrogen is also partially negative, But because of that, then the chain, bits of the chain can interact with itself, okay? And through the interaction of these hydrogen bonds. Now these hydrogen bonds can result in either a helix formation of a helical folding of the chain, which we call alpha helix, or it can result in a parallel alignment of sections of the chain, which we can call beta pleated sheet. But these are microstructures, okay? It's still kind of flopping around in the cytoplasm at this point, or in the cisternae of the rough endoplasmic reticulum, still flopping around. Even though it's got these little bits of alpha helix and little bits of beta sheet, it's still not its final three-dimensional folded structure. That's where the tertiary structure comes in. And actually, I'm going to put an asterisk here because I think most of your work should focus on understanding what primary structure is and understanding what tertiary structure is. Okay? So tertiary structure is when these microstructures fold further in upon themselves to form the final three-dimensionally folded polypeptide. Okay, 3D folded peptide. So, but why does that happen? It happens because of interactions between the R groups. It happens because of the R group interactions. Okay, and long story short, because there's 20 different types of R groups, because they all have their unique properties, some of them are hydrophilic, some of them are hydrophobic, some of them are charged, some of them are polar. Because they have these individual properties, it can result in 4 different kinds of interactions, which you need to know about. So what are the R group interactions that cause the three-dimensional folding of our polypeptide? They are disulfide, ionic, hydrogen bonding, and non-polar interactions, okay? So the side chains at this point play their role, okay? And as the side chains interact because of where they are, and because of their unique properties, it causes every protein with a unique primary structure, therefore folds in a unique way. And because it folds in a unique way, it has a unique shape. Okay? So this is really key. It is the three-dimensional folding of a polypeptide that determines its ultimate function. Okay? But another aspect of its final three-dimensional fold is that perhaps one polypeptide by itself may not be the ending of the story. Okay, some proteins, they have their final function when they are interacting with other polypeptides. Okay, as is the case for hemoglobin. So hemoglobin has one folded polypeptide, but it will only fully work when it combines with 3 other polypeptides, similarly three-dimensionally folded. But when separate, or when more than one polypeptide chain come together, that is the quaternary structure, okay? Or at least one aspect of it. Okay, so what makes up the quaternary structure? Well, when more than one polypeptide is required in the final protein, that's quaternary structure. But quaternary structure might also be prosthetic groups, okay? So these are non-protein things that are required for the protein to form its to have its function. Okay, so prosthetic groups and more than one polypeptide chain, that is quaternary structure, and that essentially then is us talking about now in the language of protein structure, these things are essential. Okay, so let's just quickly remind ourselves. So the next question is, what are the roles of proteins? Okay, so these are examples of what proteins do in the body. Okay, now we're going to divide up, based on their structures, we got 2 kinds of proteins. We've got globular proteins, we've got fibrous proteins. We'll quickly discuss what they are, and then we'll talk about their examples in the body. So globular proteins are spherical, okay? They are soluble. They've got significant three-dimensional folding, and in terms of their structure, they tend to have their hydrophilic R groups on the surface, okay? And they have their hydrophilic R groups in the core of the protein, away from the water, okay? So this is what it means to be a globular protein. Now, some of them are also, as part of their tertiary structure, they're conjugated, their quaternary structure. As part of their quaternary structure, they are conjugated. Okay, what that means is they've got a prosthetic group. They've got something that's not protein as part of their core structure. Okay, so what are the roles of globular proteins? Okay, well, the main one is enzymes, right? So, and we're going to spend a lot of time on enzymes anyway, right? But these are, yeah, they are also transport proteins, such as hemoglobin. Okay, in terms of examples of enzymes, so you can give some context to it, we are talking about things like catalase, which breaks down hydrogen peroxide. We might be talking about carbonic anhydrase, right, which we are going to talk about in module 3. Okay, but you know, enzymes are proteins that speed up chemical reactions. Antibodies, so they can have a defensive function. Other examples of globular proteins are transport proteins in the membrane. We'll call them membrane, because hemoglobin is a transport protein that's carrying molecules from carrying oxygen, carbon dioxide in the blood. But there are transport proteins in the membrane which are helping things to cross the membrane, right? So we've got membrane transport proteins. Globular proteins might also be receptors, right? Helping cells communicate or receive signals from distant parts of the body, such as hormones, or just interacting with neighboring cells and communicating with neighboring cells. So they can be receptor proteins. So these are all the kind of functions that are associated generally with globular proteins. That brings us to fibrous proteins. So fibrous proteins, well, what's the definition of a fibrous protein? A fibrous protein tends to have very little three-dimensional folding. Okay, it's got very little three-dimensional folding. Generally, they tend to be insoluble, right? So they can't float around without binding to something else. Okay, so they're insoluble. They have very little three-dimensional structure. They tend to polymerize. So they because they're insoluble, they tend to stick to each other and form these long fibrous structures, which results in their kind of contribution to the biology of the body. Okay, so what are the examples of these fibrous kinds of proteins? And the other thing that I would say here is they tend to be insoluble and they polymerize. One thing that helps them to polymerize is that they might have a greater proportion of amino acids with small R groups. Okay, so smaller R group amino acids. Okay, now because these R groups are not big and bulky, because these R groups are small, the proteins can come closer to each other and form these associations that allow them to polymerize. Okay, so that's an important feature. What are these proteins that you need to know about? Well, we have collagen, okay, we have keratin, and we have elastin, right? Now, these are significant in various, obviously they're significant in biology generally, but they are significant, they pop up in other places in our course. For example, collagen, what's collagen's main function is strength and flexibility. Strength combined with flexibility. Okay? Keratin, you could say, is mainly to do with strength. And elastin, its main property is strength, but also elasticity. So where do we find these proteins? So collagen, strength and flexibility, we find it in connective tissue. Okay, so we might find it where there needs to be flexibility and strength. For example, in blood vessels. Yeah, keratin, we find it in hair, skin, nails. All right, and elastin, again, blood vessels, but mainly the arteries. Okay, because we got the high blood pressure, and we need the ability to recoil after the stretch. After the blood has passed through, we need the recoil to maintain that blood pressure. Okay, so we see how proteins are suited to particular things, but the takeaway point of this is their structure These proteins each have unique structures, and because they have unique structures, they have unique functions. And their structures are ultimately determined by the sequence of amino acids in their primary structure. Finally then, we are going to talk about how we can investigate proteins practically. There's two main things we can investigate about proteins practically. One is their presence, okay? So one is determining the presence of proteins, but we can also investigate which amino acids make up a protein, okay? So we can investigate amino acids in terms of we can identify which amino acids make up a protein. Okay, so how do we test for the presence of proteins? Well, the presence of proteins, remember, we do the biuret test. What is a positive result for a biuret test? It is the change in color from blue to lilac or purple. Okay, and that's it really. In terms of steps, we simply add the biuret reagent. We add biuret reagent, and two, we simply observe the color change if there is one. There's no heating involved in this one. We observe the color change. Okay, if the protein is present, it should go from blue to lilac. I would add though that we can make this a quantitative test. Okay, if we are specifically looking for that lilacness, if we use a colorimeter, and we use a filter in the yellow and green part of the spectrum, we should be detecting the lilacness or the purpleness of the solution. Okay, and in that way we can see how intense that lilac color is, and we can use that to quantify, obviously using calibration curve, serial dilution, known concentrations, and calibration. By doing all of that, we can then maybe estimate how much protein is present. Okay, so that's that part of it. But we can also investigate amino acids, compositions of proteins. Okay, the basic principle of that would be thin layer chromatography. Remember what that's all about. It's about having a TLC plate, okay, and you draw a line at the bottom, you spot your known amino acids, and then you might spot some unknown amino acids using a capillary tube. Okay, you make an intense spot of those amino acids on the line. You then put that TLC plate in a beaker that contains some organic solvent. As the organic solvent rises up the solid phase, right? As the organic solvent rises up, depending on the property of each amino acid, depending on how well it will dissolve in that organic solvent, it will move up the chromatogram to different distances. Okay? Yeah? Okay? And at the end, you must catch the solvent. You must stop this process before the solvent gets right to the top. Okay? So then remember, we calculate the RF value. So for each of your known amino acids, you would calculate your RF value. Okay? And that RF value is determined by dividing the distance from the line to the spot. Yeah? Distance spot divided by distance solvent. Okay? And that should give you a value. Then you can identify what your unknown is by comparing the RF values. Or you can do this, and because each amino acid has a known RF value in a particular solvent, you can compare your unknown RF value to known values and determine which amino acids you have. Okay? You might need to go, at the beginning, you might need to digest your protein with acid to separate the amino acids, to break the peptide bonds, separate out all the amino acids, and then repeat the process to find out which amino acids are most common in a protein. Okay? But there we have it. I hope I've not forgotten anything too important. Oh yes, in order to visualize your amino acids to see where they have migrated, you may you do have to spray with a chemical called ninhydrin. And by doing that, it allows you to visualize where the spots are, and once you can observe the spots, then you can do the calculation for the RF values. Okay, so that has been proteins. Hopefully I can trim that down to not so long. Essential questions. Okay, yes, the answers could be a little bit involved, but you know, it's going to take some time for you to, if you repeat it enough, you will be able to do this. Every time you do it, you're going to get slightly better at it, and that's the whole point. So what are the essential questions for proteins? How do we describe the structure of proteins? Okay, basically it's a discussion of primary to quaternary in as much detail as you can do it. What are the roles of proteins? Being aware of where proteins come up in our specification, basically. Okay, and 3, how do we investigate proteins practically, having some experience of that, having some knowledge of that, and being able to relate the key details of that. Okay guys, this is where I finish. This is where you get to work. Good luck.